Hello guys and welcome back to Excelsior Academy. So now we are going to do the lecture 4 in which we are going to talk about the techniques which we use to carry out plant tissue culture. Okay, so keep in mind whenever you go for the next lecture, so you need to go through the previous lecture, otherwise it is not going to make any sense to you guys. Okay, so yeah, so what is the basic methodology of the technique we use in order to carry out the plant tissue culture? So first one, as you know, you need to have the media. Okay, now this media is based on the whatever plant you select. So mainly we use MS media only, that is your Murashik and Scoop media. Okay, and everything needs to be autoclave, right? Now selection of the explants, so depending on the type of plant or plant part you're going to use, you're going to take the explant. Okay, so it can be the bud, a part of leaf, a root, seed, etc. It can be any part of the plant. Okay, and then you need to sterilize. So remember from the previous lecture, how do we do the surface sterilization? By using the sodium hypochlorite or mercury chloride and wash aseptically for 6 to 10 times with the sterilized water. Okay, because we don't need the presence of these salts present whenever in further we are going to do the plant tissue culture. So we don't want this. So you need to sterilize it and then carry out the experiment. So don't worry guys, in this lecture I'm going to show you the video which of course I have taken from the YouTube itself. I'll share the site also with you guys, and we'll see how exactly the plant tissue culture technique has been is carried out. Okay, so you need to watch the video till the end. So inoculation. So once the explant is the explant is sterilized, then you are going to inoculate. That is, you're going to transfer onto the solidified media under aseptic conditions. So usually we use laminar airflow under hood. We carry out this experiment. Okay, then you keep it for the incubation. So incubation again. So you have got the transfer room which has got the controlled temperature, relative humidity of around 50 to 70%, and the photo period. So again, if you remember from the previous lecture, we have talked about the photo period. They use white luminescent or the white tube light they use. Okay. Now regeneration. So plantlets regenerated after transferring a portion of the callus into the another medium, and induction of roots and shoots. Sorry, it's not shoes, it's shoots directly from the explants. Now, how it is being done? So once you have transferred into the boiling tube, so you had the MS media, solidified MS media, you put a small part of the plant. Then you allow to grow, it becomes callus. Okay. Now from the callus, again you divide into several, and there you add the phytohormones. So you're going to add the phytohormone. So if you remember from the previous slide, so auxin phytohormone is used for the root induction, and if you're using cytokinin, then it is for the shoot induction. So in this way, you get the entire regeneration of the plants. Okay. Then hardening. So it is the gradual exposure of plantlets for the acclimatization to the environmental conditions. So once in the test tube or in the bottle, you have got the small plantlet, then you put into the pot having the soil, and then you transfer it to the land or into the greenhouse chamber. You can put it over there. Okay. Then the plantlets are transferred to greenhouse or the field conditions. So depending on the conditions of the plantlets, then the hardening and the plantlet transfer is being done. Okay. This is all very interesting actually once you do for yourself. Now let us say we have got the carrot. You break it into pieces. Okay, this is just one example. So you break it into the pieces. Okay, take that thing, put into the media. So you can see that a small portion of that has been taken and put into the media. It is allowed, it is incubated and allowed to form callus. So a callus is formed in 4 to 6 weeks. Now remember that during this time, the chances of contamination are more. Okay. So that's why you need to do everything aseptically. And as I said, if suppose you're lousy in your practicals, then you need to add antibiotics in all steps. Okay. Now, once the callus has been formed, you can add auxin and cytokinin in order for the plants to grow root and shoot. Okay. Now, that will take another 4 to 8 weeks. Then once you get the small plants, you can transfer it to the soil. And finally, what you get is the mature plant. Okay. So what you can get from here is that even a small piece can be converted into the entire plant. Now, this is a property of the plants called totipotency. I hope you know all this thing. Okay. It is the property of the plants of having the totipotency to get regenerated from a single cell also. Now, this is just another image. So you know that you have taken the plant and small pieces you have taken and then grown into the media. So you can see in the petri dish, a plant has been prepared. This is your media in the boiling tube. You can see small portion has been put over there. Then depending on the phytohormones you provide, you can see a small planted has been regenerated. Then after addition or more amount of media if you provide, it can grow into small plants. And then that can be transferred into the more bigger container, or you can transfer it to the what do you call it as the pot, okay, having the soil, or into the field directly you can do that thing. Now this is the laminar airflow. This is where you do your aseptical condition, aseptically transferring of the callus from one boiling tube to the another. This is your laminar airflow, and this is how the shells or the tissue culture rack looks like. You can see that so many bottles are there, having so many small planets. In this, they have got the controlled environmental conditions of both relative humidity, the temperature, the photo period. Everything has been controlled. Okay. So now this is the video. I'm going to explain as a video is progressing so that you can understand how exactly the plant tissue culture is being done. Okay. So let's watch the video, and I'll be doing the, I'll be also saying what all exactly is happening. So yes, you can see that the steps of plant tissue culture. So let's talk about these steps. First is the preparation of the instruments and the nutrient culture media. Of course, this also we have discussed. Sterilization of culture media using the autoclave. Preparation of explant. So you do the surface sterilization over here. Then inoculation of explant into the Murashi shoes media. Then you incubate, and then these plantlets are transferred. Okay, that is the hardening. Now let's talk about the preparation of instruments. So what are the apparatus we require for the plant tissue culture? We have got the forceps, scissors, blade holder, surgical blades. All these are required in order to hold the plant so that you can make small pieces. Okay. Now you need the apparatus also in order to make the media. Then all these are required to prepare the media. Then disposable pipette tips so that you can add the growth factors and all. Now reusable pipettes are there, bottles are there. These are glass bottles, so you can wash it after once it has been used. Okay, so you keep it everything ready. You can see everything has been autoclaved and kept ready over here. So this is how you actually do the packing of the material so that you can keep it for the autoclaving. So this is how you wrap all the forceps and the blade, surgical blade, etc. Here you must have seen the petri dishes made of glass were also wrapped. Now these are the over here, the washing is happening. This is nothing but you can just dip into the soap water, keep it for some time, and after that you can rinse it. Okay. So everything has been washed. So washing, no need to teach. Everyone knows how to do the washing. So even if you're a boy or a girl, you should know the washing. Yeah. So once it has been washed, you can see that this is the hot air oven. It is kept for the drying, and later it will be wrapped and again autoclaved. So now your instruments of the apparatus has been ready. Now coming to the culture medium. So exactly you're going to prepare your media for the regeneration of the plants. You can see so many chemicals are present. All right. So we know that macro elements and micro elements are required as we discussed in the previous lecture. So sucrose as the carbon source, you're going to weigh. So many people either they do it like this, or you can take a piece of paper. Onto that you can also measure the amount required in order to make the media. Okay, so different people are having different approach. Either you can do it on the paper, or you can have a plastic container. So whenever you're adding, just keep the double distilled water onto stirring so that immediately it gets dissolved. Then, of course, before autoclaving, you need to adjust the pH of the media, as again discussed in the earlier lecture. So depending on that, you have to adjust the pH around 5.6 to 5.8 or 6 we have just discussed. So you can use HCl and NaOH in order to adjust the pH. So these are the conical flowers which you're going to need it later. So wash it, and dry it and then sterilize it, okay, using the autoclave. So this is how they are making the cotton plug. Now, how to do the sterilization? That is with the help of the autoclave. So these are very big autoclaves actually. So at a time, many of the containers you can keep it. Okay. So usually assistants will be there. If not, then you have to do yourself. So you know the temperature is around 121 degrees Celsius and pressure is around 50, and we keep it for 15 minutes. Now, okay, so you have just adjusted. Now these are the explants. It can be leaves, flower buds, ovaries, seeds, stamens, anthers, anything it can be. Okay. So any portion of the plant can act as your explant. Now, so this is how you harvest the plant so that you can, make the explants later. So you keep it in the sterilized water, and then with the help of the scissors, you're going to make small pieces so that you can, make small pieces and you can transfer it to the culture media then. So now what they're doing is the surface sterilization. So they have added with the help of the sodium hypochlorite. They're just, cleaning the surface of these and then putting into the solutions of water. And then after that, you're going to wash it around 4 to 6 times with the double distilled water. Okay. So now washing has been done. Now inoculation of the explant. So now your explant is ready. Now what we are going to do, we're going to the inoculation chamber. So of course you need to change over there. Everything has to be, the sterilized condition is there. So that is your laminar airflow is kept over there. You go inside and then keep your UV ready, everything, all that what we have discussed. So now you keep the UV ready, keep it for around 15 to 20 minutes. UV has been on. Okay, now this will help in the sterilization of the chamber. And then once this has been done, then you can proceed. Okay. Yeah. So now the hand sterilization, as I mentioned, with the 70% alcohol, you have to sterilize your hands also. Then whatever the space you're working on, again, even that needs to be sterilized. So the rim of the conical plants, you need to sterilize it. Okay, with the hints and burner, mix it. This is the washing of the plant that's happening. Yeah. Okay. So this is like the media, everything you need to heat sterilize it. So that because at the rim of this, there are chances that the bacteria might be present. So again, washing is being carried out. So 2-3 times or maybe 4 to 6 times you need to do it. Okay. Then drain it, take the explant, dry it. This is the Whatman filter paper, which has been sterilized in the autoclave. Okay. So again, every time you need to show it. Then with the help of the sterilized blade, you're going to make small pieces of the stem which you have taken, which you have sterilized it. Now, these are the media which you have prepared. Okay, solid media it is there. Now, label it first. Every time keep your marker ready because you never know when you need your marker. So keep your marker ready. Don't share it. Put the date also, and you need to put your name also. So to the agar media, insert the inoculum. And then take that, put into the incubator. So you can see that over here, entire room has been, having the control environment. So either you can have these bottles or the conical flasks, or you can have the boiling tubes also. You can see. And then every time you need to check the growth of the plantlets. Okay. So maybe in 24 hours or 48 hours, at least once you have to come and see how beautiful you can see the small plantlets have been grown from one single small piece of the stem. So that is here you can see the roots are also there. Okay. So as I mentioned, roots means you need to provide oxygen, shoots, all these leaves, you need to provide cytokinin and the other regulators also. Yeah, then after that, once these has been made, you need to start the hardening. Now, what is hardening? You need to start putting into the soil. So on these pots, you can transfer it. And after that, you can transfer it to the landfills also. Okay, so everything, the carbon monoxide, airflow, shock, temperature, humidity, everything you need to control. So how beautiful you can see how many plantlets have been growing now from one single small seed. So this is how you do the hardening of the plantlets. You take it out from the media and then put it into the soil. And each pot you need to label it so that you can have the track of all these things. Okay. So all these you need to add, because manure also you need to add so that the plants can grow properly. And after this, you can put it into the landfills. So this is how beautifully you can get your land full of these plant beds. So that's all for today. Hope you guys understood how exactly we carry out the plant tissue culture, and all these we are going to study in detail. So this was only one thing, one basic thing. Later we are going to talk about different types of culture. Like here, what do you mean by micro propagation? How ovariculture, anther culture, nucellus culture, embryo culture, all these being carried out. Okay, so all these we'll be discussing slowly in the upcoming lectures. So thank you guys for listening this and watching the video. If you like the video, please share and subscribe with your friend circle also. And yeah, thank you for watching.